2, 1.
Good morning, everyone. Welcome to day 3 of the HypoPARAthyroidism Association. A few housekeeping rules. Please note that your microphone is muted, but if you have any questions, please utilize the chat function on the right hand of your screen. So good morning, I am Barry, and I am the treasurer of the HypoPARAthyroidism Association. I am also the face behind our Instagram and Twitter accounts and one of the Facebook faces. So please make sure you follow us if you use those applications. I know at times we feel isolated and alone. These platforms will help you know that we are a community and we are all here for each other. I graduated from the University of Delaware in 1995 with a Bachelor of Science in Business Administration with a concentration in marketing. I was diagnosed with hypoparathyroidism in 1998 after a total thyroidectomy. It's been a very long and rocky road, but I'm so grateful to be here today. I am currently on the TransCon clinical trial, and I feel amazing. I never thought I'd be able to say those words. Someone recently asked how I felt now that I was on this medication as compared to my previous treatment plans. I replied, it's like drinking a Mountain Dew. I feel like I can climb mountains. I would like to thank you all for being here with us this weekend. We have over 500 attendees from 22 countries worldwide. To say this is our biggest conference yet is an understatement. We have far exceeded any goals we had set. I know it's been a complex and difficult time for most of us, but then again, we are rare. So if anyone gets complicated or difficult, it is definitely us. I am happy that we have each other to lean on, although it is virtually. I always love reading advice and stories on our community Facebook page. We are like a family who shares milestones, triumphs, tragedies, and just some good advice, whether it's a recipe, parenting, clinical trial information, or just knowing that someone out there actually gets it. We are all different people with the same common denominator, and I am so happy that we are all here together, even if we are hiding behind a computer screen. I know it hasn't been easy, but we are so grateful to have all of you here today. So thank you to all of us for joining us, whether you are just waking up or just going to bed. We would love to be together talking and telling stories about what has happened in our life this past year, but we cannot. The sad fact is more people have died from COVID-19 in the United States than the number of people diagnosed with hypoparathyroidism worldwide. I like numbers, so I tried to do some math, and please do not quote me on this, but I figure approximately 1 person for every 38,000 people in the world are diagnosed with hypoparathyroidism. We are indeed a rare breed. We hope to be together next year, sharing our stories and hugging and catching up. But for now, we are here virtually, and for that, I am thankful. I would first like to thank our board of directors. We are scattered across the United States, all battling hypopara, yet we were able to pull this off successfully. Thank you to Chairman of the Board, Bob Sanders. Your speech the other day was incredible. I'm sure I'm not the only one who had to grab a tissue. I would like to thank Vice Chairman of the Board, President Deborah Murphy. You are amazing, and this never would be possible without you, and we all love you. I'd like to thank Loretta Gully, who is our secretary, and she has been so busy taking minutes for every one of our meetings, which has been a lot, and we couldn't do without you. I want to thank Joellen Friedenberg, who is responsible for maintaining our amazing new website. It is a work in progress, and it is just amazing all the work she's doing for us. And I'd like to thank Michelle Reyes, who has done an incredible job running the silent auction. We raised $732, and we are so very, very proud of her hard work. And I would like to thank Patty Keating, who took a brief hiatus from the board, but came back, and we are so happy to have you. You are incredible, and I don't know what we'd do without you. So thank you, guys. It's a privilege and an honor to serve on this board with you. I would next like to thank our doctors. You have all provided us with so much information and resources. You are amazing, and what you do for our rare and mighty group is extraordinary. Thank you for taking time to share your wisdom and dedicating yourselves to helping our community. We are grateful for all of you. Thank you. This conference never would have been possible without our sponsors. A big, big thank you to Amylite, Ascendis, Calisthenics, Takeda, and the HypoPARA Research Foundation. Your dedication and research you do on behalf of our rare community is remarkable. Thank you for dedicating so much time and energy to help us fight this disease. You're finally giving us some options instead of the standard treatment of care, which, as you know, is not perfect. We are grateful for all of you. Thank you. I would also like to thank Amanda from Skill Me Up. This platform is amazing. You have worked so hard to get this conference up and running. Thank you. You have been there for us 24 hours a day, 7 days a week for the past few months. And we appreciate all you have done to make this conference such a huge success. With the overwhelming success of this conference, we have decided to pave the way for our future. Hopefully, we will be able to meet in person next year, but offer a virtual platform as well. So wherever you are in the world, you can stay up to date with HypoPARA news and resources. There are so many new and exciting treatment options for us. It is nice that we will finally have an option. And here is where we get to share it with you. We are thrilled to have people from all over the world. Next year, we hope to have our conference in Los Angeles, California, and be virtual as well. So if we can't travel because of health or other reasons, we can still be together. This was a trial run and a successful one at that. We offered it for free because, quite honestly, we didn't know if we could pull it off. But here we are at day 3, and we are doing great. In the future, there will be a charge, as were in our previous conferences over the last 2 decades. Our board of directors is completely voluntary. We work across time zones to help all of you, all of us. We are hoping that you can find it in your heart to make a donation to the HypoPARAthyroidism Association. All monies go towards future research and development. We carry little to no operational costs because, as I said, we are volunteers. We offer monthly giving as a way to help our association. If each attendee donated $10 a month, the HypoPARAthyroidism Association would receive $60,000. If each donated $20 a month, we would raise $120,000. Monthly giving is a way for us to sustain and grow. You can sign up for monthly giving on our website under the How to Help tab and then donate. There's a box you can check off for monthly payments. And if each of you donated the cost of last year's conference and banquet of $100 today, the HypoPARAthyroidism Association would raise $50,000. The information we learned these past few days is so valuable. We want to continue to grow and offer you all of this and more. All of the money we raise will go towards helping education. We have upcoming webinars on various topics, including dental care, caregiving, living with HypoPARA, as well as nutrition and holistic options. We would also like to have small in-person regional group meetings around the country, similar to ones we've done in the past. We are also working on translating our information into other languages. The money will also go towards continuing training of our board so we can stay relevant in today's digital world. You can also donate by simply shopping through Amazon Smile. You can refer to our website for more information, and you can donate directly through there. I know it's a lot to ask during these trying times, but your small tax-deductible donation will be put to work. Our mission is to improve the lives of people impacted by hypoparathyroidism through education, support, research, and advocacy. Your help today would bring us to the next level. Our growth is dependent on our members. I hope you can find it in your heart to help us achieve our goals of growing. We welcome whatever you feel comfortable giving. You can find a link on hypopara.org. Thank you in advance. We love and appreciate all of you. We hope everyone has enjoyed the conference so far. It's been so much information and it's really been wonderful. We appreciate all of the feedback and we are excited to see what our future looks like. So thank you, everyone. Now, speaking of the future, I am going to introduce our first speaker of the day. Our first speaker is Dr. Diane Krause. She's originally from Pittsburgh, Pennsylvania, and received a degree in biology from Brown University. She then continued her studies at the University of Pennsylvania, where she received both an MD and a PhD. After completing a residency in clinical pathology at the University of Pennsylvania, she performed postdoctoral studies at Johns Hopkins University. Dr. Krause is presently professor of laboratory medicine, pathology, and cell biology at Yale University, associate director of the Yale Stem Cell Cancer Center, associate director of transfusion medicine, and director of the Clinical Cell Processing Laboratory, which collects and processes cells from bone marrow transplantation. She runs the well-funding and expanding research laboratory focusing on leukemia, stem cells, clinical cell therapy, and pluripotent stem cell differentiation down to the parathyroid lineage. So please, let's give our attention to Dr. Krause, who will be speaking about our future. And thank you, everyone.
Dr. Krause, you're great to start.
Hello, everybody. I hope that you can see me and hear me. I first want to thank you very much for the honor of being invited to speak at this wonderful conference. This is my first time attending, and it's been phenomenal. I look forward to attending in person in the future so that I can meet each and every one of you, because I think that that would be super exciting and fun. So this will be my first introduction to the group, and feel free to ask questions throughout. I also have some stop points throughout where I just pause and ask if there are any pending questions. So my goal is to tell you about our work to use stem cells for a treatment of hypoparathyroidism. So as Barry mentioned to you, I have in the past studied leukemia and bone marrow stem cells, how blood forms. And I also have studied how you can use cells to treat patients. And usually we use blood forming stem cells. So you've heard of bone marrow transplantation. So it was when I met Julianne Sosa, who is an endocrine surgeon. Many of you may know her. She's now head of surgery at UCSF. And she was my neighbor across the street. She said, you know, you do stem cell research. I think you should look at parathyroids. And that was the beginning of this about 8 or 9 years ago. So we've made some progress since then, and I'm happy to tell you about it. I can get this to work. I'm having some trouble getting the slides to move.
Okay.
Oh, there you go.
There you
Okay, so why focus on cell therapy for hypoparathyroidism? Well, obviously we want a cure for this disease, and we've heard so much across this conference about people who are developing wonderful novel treatments, and I think that the drug developments have been hugely exciting. What could be better about stem cell treatment would be that it's permanent and does not require you to have an injection or a pill, and would actually create it so that you have functional parathyroid glands again. That's the goal. So why is it that we think that stem cell technology will work for hypopara? So I have this kind of gruesome slide, but it's meant to show what's currently done for some patients who get what we call autologous, or their own parathyroid cells re-injected into their arm. And the way this works is that when the surgeon is in the operating room, they take the parathyroid gland and they chop it up in the operating room into tiny little pieces. And then they take those little pieces and they can embed them into a muscle. And that can be in the arm, it can be in the neck. And these cells actually function long-term to provide parathyroid hormone secretion in response to calcium, just the way they would when they're in the normal spot in the neck. And one of the reasons we know that this works is that we can actually, instead of putting it directly into the patient in the operating room, they can freeze it away, and then only if the patient needs it, if the patient develops hypopara afterwards, can we thaw those little bits and pieces that we've cryopreserved and then have them embedded and have them function. So the idea with stem cell therapy is a lot of patients do not have parathyroid glands that have been cryopreserved to use for their own transplantation. So we think that it's really low-hanging fruit. If we can get stem cells to actually make functional parathyroid cells, we do know that if you inject them into the muscle, that they should work. So the other caveat here is anybody you know who had their parathyroid glands put into the muscle didn't have the situation that many of you have, where it was, for example, a total thyroidectomy that inadvertently or without being able to resolve it, where the parathyroids were because they were embedded in the thyroid. These patients didn't have that. These patients had hyperparathyroidism, secondary hyperparathyroidism, where they had overgrowth of the thyroid, and they were having that removed. And that's what's been used for transplant historically, not normal parathyroid tissue. Okay, so this is a slide that's meant to show you about what pluripotent stem cells can do and why we use them. So pluripotent cells, pluri means all or many, and potency, it can become all cell types in the body. So a pluripotent stem cell, the original ones were derived from actual embryos. So, you know, the sperm and the egg fuse, you get a zygote, and you get this very, very small embryo, and it forms this circle of cells. And within that is this ICM, it doesn't matter. And these cells are the ones that are going to become the actual fetus, and the rest are just supporting it. These cells can be grown in vitro and make a cell line that we call an embryonic stem cell line. And just like the normal embryo, which can make every cell type in the body, these cells, when they're grown in vitro, can be coaxed to become different cell types. But the only way to do that is if you know what happens in the actual embryo, so you can reproduce that series of events to get the cells to differentiate. And nobody has yet been able to get these cells to differentiate in vitro into functional parathyroid cells. So that's our goal. And this is what these cells look like when they're grown in vitro. Maybe this will work, but maybe it won't. Oh, there's supposed to be a video here. But anyway, when you grow them in vitro and let them differentiate, they actually can start beating like hearts, but unfortunately that isn't going to work. So here's the goal. The goal is from the patient with hypopara, we collect some cells. We can just collect blood, or we could take a skin biopsy. And what we would do is reprogram those cells to pluripotency, or make induced pluripotent stem cells. And this is work that you heard about on Friday from Dr. Michael Levine, and he and I are working in collaboration on this work. So if you reprogram these cells to pluripotency, then here's the trick, and this is where my lab is focused. You would then, through a series of steps, differentiate them to functional parathyroid cells. That's these little purple dots. We want to be able to isolate out those parathyroid cells, prove that all we have is parathyroid cells, because these cells are tumorigenic if you put in the undifferentiated cells. And then take these very safe parathyroid producing cells and transplant them back into the patient, in the muscle, and have them respond long-term and cure hypopara. That's the goal. So we think this is going to work. Like we already know that you can take functional parathyroid cells and transplant them in the operating room. And secondly, for diabetes, this has been in the works for about 15 years and is starting to get closer to the clinic. So what people have done is taken these pluripotent stem cells and made pancreatic beta cells, the ones that make insulin, in vitro. They've been able to reproduce their clustering. And here's a nice picture of these pancreatic beta cells that are being formed by a company, ViaCyte, that hopes to get to clinical trials where they're injecting these into patients. Also, these cells have been injected into patients for macular degeneration, which is a form of age-related blindness. It also occurs genetically in some people when they're younger. So the back of the eye is another cell type called retinal pigment epithelial cells. And people have been able to make retinal pigment epithelial cells. So these are two that have progressed, and we're hoping parathyroid will not only catch up, but surpass them. So there are a lot of scientific challenges. First of all, we need to recapitulate normal embryonic development in a dish. And unfortunately, we don't fully understand how parathyroids form in the embryo. It's really understudied. So that's one of the things that we really have to do, is figure out every step of the way of what happens to form parathyroid cells during fetal development. We don't have good parathyroid cell lines for controls. These are not cells that grow well in vitro for us to study. We also don't have normal parathyroid tissue for controls because we can't take patients' parathyroid cells, parathyroid glands out when they're healthy. And we don't have very many ways of knowing whether or not we've identified a parathyroid cell, because you have to have a way of adding an antibody to the outside of the cell against something that's specific for that cell so you can label the cell. And the only thing we have right now is your calcium sensing receptor. The parathyroid hormone's on the inside of the cell. You can't see that. But on the outside of the cell, you can add something. So we need to do something better than the calcium sensing receptor, because while that is on all of your parathyroid cells, it's not on only parathyroid cells. There are a lot of other cells that have calcium sensing receptors in our bodies. Okay, so I'm not going to go through this in detail, but the point is, during embryogenesis, there are many, many steps. There's the mesoderm forms, the endoderm, the foregut. Then you get to this anterior and posterior foregut. The posterior foregut is well studied in cell development, and that's because that's where the pancreas is. So people have been studying pancreatic beta cell development. But up here where you have the thyroid and the lung and the parathyroids needs to be studied much more deeply. So what we're doing is we're taking a developmental approach and trying to reproduce each step in vitro from these undifferentiated embryonic stem cells. So this is just a bunch of letters, but it's actually our map for what we need to do to make these cells become parathyroid cells. So what we know is we have to start out with undifferentiated cells that express these genes, OCT4, NANOG, and SOX2. We then need to get them to this orange stage so that they have this gene expression pattern. Then we get them to this gray one. Then we get them to these green ones. And then eventually we get to the purple ones where you can see PTH, parathyroid hormone. That would be the cells that are making parathyroid hormone. And that's what we've now succeeded in doing. And I'll show you some of those data. So first, Betty Lawton, who is an incredible scientist who's been working in my laboratory on this for 7 years, had to optimize the very first step, how you get the cells to make endoderm. And she published that in 2013. Then another laboratory, that of Hans Snook, optimized how to get the cells to the next step, to the anterior foregut. And then we had more steps, many, many more steps that we needed to take to get to cells that are making parathyroid hormone. So this is the first step that I was telling you about that Betty published in 2013, where she ended up having to play around with extracellular materials that support the cell. You know, these cells are actually forming in vivo in an environment that is very rich in lots and lots of nutrients. And she discovered that if she adds these nutrients on the top and on the bottom in a semi-solid matrix, that the cells do much, much better. We get much better gene expression, and we get much better cell survival. So that was step one. Then she took many, many other steps. So now we'll move on. I'm just going to stop here for questions. Barry, any questions? No?
Okay, hi, sorry, my microphone is muted. Well, someone wanted to know, 1 second, let me just go right back there. With a transplant, though, you will need to be on heavy-duty and anti-rejection meds for the rest of your life. Is that correct?
That is such a good question. Ideally, what we would do is have autologous or your own cells. In that case, you would not need to take any medications for rejection. So that would be the ideal.
Wow.
What ends up happening, and this is very likely, it's certainly going to be where it starts, that there is a bank of cells that work, but they're not actually from the patient. Then yes, there would need to be anti-rejection meds, and that wouldn't be ideal because the long-term cure would require that you be immunosuppressed. The only limitation really to having autologous cells rather than this bank of other cells is cost. Because if we have to make the cells for each person, that's going to be much more expensive than if you have a bank of cells that you can use basically as a drug. What the companies are doing, at least for diabetes, and I think we can take advantage of this for our work, is they're making this bank of cells to be a bank of cells that would be rejected by nobody. They're trying to modify it so that the genes that are on those cells that would be recognized as foreign won't be there. So they're making additional improvements in this. But if we did it right now, we would definitely need to use anti-rejection meds as long as it's allogeneic, somebody other than the patient.
Okay, now is stem cell treatment an option for a person with the CASR gene mutation?
Oh, that's a really good question. The answer would be no, because if your calcium sensing receptor isn't working, your parathyroid hormone release is actually normal. So I think that in that case, actually, I think it would be helpful in some respects. If you got third-party cells, not your own cells, because your own cells have a mutant CASR. If you got third-party cells, then at least your parathyroid would be releasing parathyroid hormone correctly in response to low calcium. The problem is that calcium sensing receptor has to function throughout the body, and it wouldn't be correcting those other cell types, but it would be correcting your parathyroid to be secreting the parathyroid hormone appropriately. So it's a partial correction.
And now, would having a non-functioning reimplanted morcellated parathyroid tissue from a parathyroid adenoma help or hinder eligibility for this process?
Oh, I don't think that would make you ineligible. If you had the transplant and it didn't work, then you still need functional parathyroid. So I think that would just be that this would be much, much better. Perhaps for some reason that didn't take or didn't function after cryopreservation. I don't know your particular situation, but no, it would not be something that prevented this from functioning.
Okay, now I'm going to ask one more question, then let you get back to your presentation. When daughter gives birth, should embryonic cells, umbilical cord be saved? And could this be used for future stems for myself to make parathyroid cells?
Okay, so should we be cryopreserving cord blood for this purpose? The answer is not for this purpose. That wouldn't necessarily be any reason to do it, because your offspring's cord blood is not the same as autologous, so it would still be a third party. Maybe if we actually cryopreserved cord blood for 50 years and everybody had it, which isn't how it works. It could be that if I had HypoPARA because my thyroid was removed, but there were some cryopreserved cord blood cells, we could use those. But they're no better really than the cells I have in my body, because either way we have to reprogram them back to pluripotency. The ones that are in the cord blood are not embryonic stem cells, they're not pluripotent. So I would say no. Short answer,
Okay.
no.
All right, so continue, and then there are more questions that keep coming in, but I want you to make sure you get your full presentation.
All right, so moving on to the next step, telling you about the fact that we now have PTH expression. So one of the first things that Betty did, and I'm not going to go through all these steps, but she had to play for 38 days. She made some different changes in the cells in vitro and took multiple steps. So we knew these 2 steps, but she had to figure out all of these. And this was an experiment where we're looking at the gene expression for a bunch of genes that we expect to see in parathyroid cells. For example, CASR and GCM2. But this was super exciting. This is the first time that we saw parathyroid hormone. So we were very excited about that. And then we made further improvements to the protocol. And we were able to get parathyroid hormone repeatedly. Then we did something that I'm super excited about. We contacted Mike Levine's lab and we started collaborating. And the reason we did that is so many people who work on pluripotent stem cells get a protocol to work in their lab. It's legitimate, it's working. But when they give the protocol to other people who do it in their own labs, it doesn't work. And we don't know why. So we gave our protocol to Mike Levine. And his lab not only repeated our protocol, they got better data than we did. So this is phenomenal. So that's why I'm showing you his data here. And he showed you this in his talk as well. So this is the CHOP induced pluripotent stem cell line that he has in his laboratory. And what he did is he differentiated the cells for 36 days. And this is the amount of GCM2, which we know is necessary for parathyroid cells to develop. You see that it's expressed here before you get to the parathyroid cells. And then he also looked at parathyroid hormone expression. And what you can see is they got a 102,000-fold increase in parathyroid expression compared to when they started. So this was super exciting, and we ended up publishing this work. So this is the currently optimized protocol. As I told you, it's a 37-day protocol. These are all the different things that Betty had optimized in vitro to get to parathyroid hormone expressing cells. And this was the paper that was published in October of 2020 with Michael Levine's work lab. And I should mention, Corinne Martineau in Michael Levine's lab really did those beautiful experiments I just showed you with the huge amount of parathyroid hormone being expressed. So that's great. We're super excited. This has been so hard to achieve, but it's actually only one step, and we need to take many more steps. What we have, everybody has the same DNA in all of their cells. We all have the gene for parathyroid hormone, but it's off. And we had to figure out how to turn it on. So we turned on the gene and we saw RNA. That's what I was showing you in this previous slide. So this is RNA for parathyroid hormone. But what we really need is parathyroid protein. And wait, sorry. On Sunday morning. Okay, so what we need is parathyroid hormone, the actual protein, and to prove that these cells are functional. And that is where we are right now. Now, why aren't we there yet? Well, we're not there yet because there's other science that needs to be optimized first, although we will keep going. I actually put in questions here as well. You want to add any, Barry?
Oh, yes. Okay. So, is there a place my doctor can find clinical data from your stem cell research?
Yes, it is published. But
it is published.
It is.
Okay.
I can send out the reference.
Okay, thank you. We'll put that link to that on our Facebook page. Also, will this process work in patients with autoimmune PT dysfunction?
No. The same thing with diabetes. Patients with diabetes, if you just gave them pancreatic beta cells, that wouldn't work because they have an autoimmune reaction against their own beta cells. So what we would need to do is immunosuppress you so you don't reject your own parathyroid hormone and then give this back. So there are two problems in that case. You know, your autoimmunity destroyed the tissue and you don't have it. So to treat both diabetes or hypopara, you need to return the functional cells and prevent the autoimmunity. So there are two challenges. And they're really being addressed by different teams of scientists. There are scientists who are working on tolerization of patients so that you can tolerate something that you have an antibody against.
Okay, now, could I get the transplant take with me if all my parathyroids are intact but have died? I don't know, does that make sense?
I'm not fully understanding it, but if your parathyroids are no longer alive, then they're not going to work in a transplant. So you would make this instead.
If a small amount of parathyroid tissue was inadvertently removed from a surgery that took place a couple of years ago, could that have been preserved, and could that be used to generate stem cells to replace my own parathyroid function?
That's a bit of an interesting question. So if your functional parathyroid that was inadvertently removed had been cryopreserved, you wouldn't even need stem cells. We would just thaw that and give it to you. The thing is, you have to realize that some of you, I don't know, everybody's different, but if your parathyroids were removed because it was embedded within a thyroid cancer, then you don't want those cells back, because at this point, we don't have the ability to perfectly separate out functional parathyroid cells that are not cancer from your thyroid cancer. We definitely don't want to put the thyroid cancer back into you. As awful as hypopara is, you don't want thyroid cancer. So it would depend on the situation. If your functional parathyroid glands had been cryopreserved and they're free of cancer, yes, they could be thawed and put back if that had been done at the right time. But sometimes the surgeons don't see the parathyroids. They're just embedded within the thyroid. It's not a mistake when they come out. It's just that it was unavoidable.
Okay. And those are all the questions I have for you right now.
Okay, so moving on. So what I told you is we need to develop functional parathyroid cells. Right now we have parathyroid hormone RNA. So one of the things you need to know is that parathyroid glands are made up of many, many cell types. And I think not only do we need to learn more about how they formed in vitro, we need to learn about these various cell types really well so that we know which ones we have to make in vitro. So amongst these cell types, you have the ones that make parathyroid hormone. Those are your chief cells and your oxyphils. There are blood vessels in there, and there are adipocytes, etc. So when that tissue is morcellated and put into the muscle, it's actually still got all of these different cell types in it. And what we don't know is which combination of cell types we need to give. Right now, if we gave just the parathyroid hormone expressing cells, that might not be enough. Maybe you also need to give these other cell types. So we're testing them. So first we wanted to know, what are all these different cell types? And there's something called single cell RNA sequencing. So what we can do is we can take parathyroid tissue, in this case parathyroid adenoma, digest it into single cells. So this isn't just morcellating the tissue. Every single cell is loose in a solution. And for every single one of those cells, find out what the RNA expression is. And then using computational biology, develop these trees that organize the cells and tell us the different cell types. And that's what I'm showing you here. This is actually super exciting data. What we've done is we have two different parathyroid adenomas from two different patients that were digested to single cells. Every cell was sequenced and then analyzed bioinformatically. And what they were able to show is, ignore all the gray cells. The gray cells here are not making parathyroid hormone. But these green, red, and orange cells were all making parathyroid hormone. And what you can see is within these populations, and there's purple as well, there are 4 different populations of cells that are making parathyroid hormone. And we're super excited about this because we think that one of them is the oxyphils, one of them is the chief cells, and that this red population might represent a precursor cell that forms both. So we might have cells within our parathyroid glands that serve as progenitors for more cells. And I think if we understand those cells even better, we're going to have identified new biology of the parathyroid gland and figured out exactly the cells we want to make from the stem cells. I think these are the ones we want to make because these are the ones that can probably make more of the chief cells and the oxyphil cells. So now we need to figure out ways of identifying those cells with the surface markers, and we're starting to do that. So we take what we learned, we've taken other data that was published on all the different genes that are expressed to identify these novel markers, And just to tell you that we can, if this is the control and this is a parathyroid, we're able to come up with ways of marking these cells. So there's nothing here. When you add this antibody, all of these cells show up. So we have some markers of parathyroid cells that we're hoping to use to identify them when we make them in vitro.
Now, I do have a question here. Okay, it is, I had an adenoma, as I think you pronounce it, in 1999. I was supposed to have any pieces of paratissue put into my forearm. They did not do it. And one doctor said because I had an adenoma, I would always be hypopara. Would transplant work?
Yes, transplant would work, and I'm sorry that they didn't save some of that adenoma. So there's adenocarcinoma and there's adenoma. So let me just make sure that we clarify this. A parathyroid adenoma, which is probably what you had, is not malignant, it's not cancer. So when we put that into the arm, we're not putting in a cancer. It's an overgrowth, but it's not cancer. In contrast, if it was a carcinoma, they would never put that back in, because that is actual malignant disease. So, but yes, this is, you would need this if you didn't get your adenoma cryopreserved to be put back into your arm.
Okay, now, someone would like you to just go back to the previous slide again. I don't know which one, but the previous. And that while they are looking at it, I will ask you another question.
I don't know which slide.
She just said the who
okay,
this person just asked for the prior slide. I still have a small amount of parathyroid function. Would it be possible to take them and create stem cells that could fully restore my parathyroid glands? Or would you need cells that have been saved from my original surgery?
The answer is we don't know yet how to expand normal parathyroid cells to make more parathyroid cells from your existing ones. So I'm glad you still have some residual function, but at this point, we don't have any way of using those cells to make more cells. If your body can't figure it out, we haven't figured it out. But I'm hoping from these single cell studies that we've done, where we see this potential progenitor population, that with that knowledge, we can in the future figure out how to get those cells to expand and make more functional cells. But at this point, no, there's nothing we can do for you, unfortunately.
I have just one or two more questions here, but first I wanted to let you know that that person's adenoma was a lipoadenoma, fatty parathyroid. Could that be used?
Theoretically, yes. I mean, I don't know the exact situation, but it doesn't sound like cancer.
Okay, and it was the correct slide. And can you take the cells from reimplanted failed parathyroid in my arm muscle to coax into a fully functioning parathyroid and reimplant? And that'll be our final question.
I don't think so. I think if your body can't get those cells to function, we're not going to be able to get those cells to function. And so that again gets back to our ability to reprogram these cells to pluripotency and then make functional parathyroid cells would be the better bet in that situation. Because your body knows best how to make parathyroid glands. And if it can't figure it out, we're not going to figure it out in the test tube.
Right. And those are all of the questions that I have here.
Moving on to our now super exciting work on functional studies. So as you can imagine, once we get these embryonic stem cells or pluripotent stem cells to make parathyroid hormone producing cells in vitro, we have to prove whether they function in vivo. And that means we need to develop good in vivo assays. So even though we're not far enough yet along with the embryonic stem cell work to put those in vivo, because first we have to get protein and have surface markers for them, we're developing our assays for functional parathyroid in animals. So that's what I'm going to tell you about now. So we need to, as I told you earlier, need to figure out which cell types we actually want to put in. And in order to figure out which cell types we have to put in, we have to have a good assay to test this. So we developed a xenogeneic, that means two different organisms. So we're putting human cells into immunodeficient mice, assay of parathyroid function. And we're testing different subpopulations of primary parathyroid, again, using adenoma tissue, because that's what's available to us. So what we do is we get the adenoma, we morcellate it into little pieces, And then we take out single cells and we sort them. We have the oxyphil cells, the chief cells, and other cell types. We put them into mice, and then after 1 or 2 months, we go back in and we find those cells, and we make sure, first we treat the mice to make them hypocalcemic. Just like when you guys have low calcium, if you have a normal parathyroid gland, it should make PTH. So we test that the parathyroid glands can make PTH and analyze the tissues. So I'm going to show you the tissues first. So what we do is we inject these cells into the muscle of the mouse. So we can go right back to the muscle and look at it under the microscope. So this is 2 different mice, tissue from 2 different mice. And what you can see, we've stained here for human tissue. So we know these are the human cells we put in. We stain for parathyroid hormone, the protein. And you can see that we have a little patch of parathyroid hormone producing cells. And here is another place. You can even see where the needle went in, where we injected the cells. They're making parathyroid hormone. This is the 2 together, the nucleus. And this is what it looked like in vitro in the muscle. So is this functioning? And the answer is yes. So I'm not going to go through this whole experiment, but there's just a few mice per group, 3 mice, 2 mice. And what we gave them is just oxyphil cells, just chief cells, a combination of all of the cells that were in the gland, and we gave them different doses. And what we did is we made the cells made the animals hypocalcemic, and then we just took some blood and we measured the parathyroid hormone in their blood, the human parathyroid hormone in their blood. And what you can see is these mice didn't have any parathyroid hormone, the ones that just got oxyphils, the ones that just got a few chief cells, no. When they got 105,000 in a combination of cell types, that's when we started seeing physiologically relevant levels of parathyroid hormone that was present in the serum after hypocalcemia. And we also saw that when we gave just chief cells. So this is a tiny little experiment. It's something we need to do many, many more times, and this is part of the ongoing work in the laboratory. So this is just showing you the same data. I didn't mean to show you that on the tiny slide. So we're able to see this. For some reason, when we gave 350,000 cells, we didn't get as good secretion as 105,000. And if you're used to looking at scientific data like this, it's actually somewhat normal to get a lot of variability in your data. We obviously need more mice. So this is in summary of the talk that I've given. We have optimized parathyroid stem cell differentiation to cells that express PTH RNA, but not protein. We've used single cell sequencing to better characterize these primary being direct from a patient, parathyroid cells, and I'm starting to identify surface markers for parathyroid hormone producing cells so that we can identify them and we can purify them. Those are those antibodies against these surface markers we can use as ways of grabbing onto the cells and purifying them. And we've transplanted these primary human cells into immunodeficient mice and proven that they're functional and that they secrete parathyroid hormone in response to hypocalcemia. So what are the next steps? Obviously, we have to increase the efficiency of our differentiation process and the number of parathyroid hormone producing cells that we have. We need to get calcium responsive PTH protein release. That's our next goal, and that's going to be huge. We need to prove that these PTH cells are then functional using our animal model. And we're going to continue to use the single cell sequencing to elucidate the molecular pathways that are important for the development of parathyroid. And we're going to do that by following parathyroid development using mice murine embryos. And we're collaborating with investigators at Yale to do that. And I continue to identify surface markers for identification and purification. These are the folks who have contributed to the work. It's been a lot of people. I did mention Julianne Sosa and Sanciana Roman, who were surgeons here at Yale and are now at UCSF, along with their colleague James Coe, who does beautiful work on parathyroid hormone and how it's released. As you know, Michael Levine and his postdoc Corinne Martineau did a huge amount of the work that was in our paper. And we have to thank Courtney Gibson, who is a fantastic endocrine surgeon here at Yale, and Betty Lawton, who did really most of the work along with help with other members of the laboratory, other surgeons. Smita Krishnaswamy is the one who did all of the bioinformatic, the computational analysis, and of course the Yale Stem Cell Center. Thank you.
Well, thank you. I have a few questions here for you. One, Does your research hold any hope for patients with idiopathic hypoparathyroidism?
Well, the thing with idiopathic is that we don't know what causes it. So the answer would be some patients with idiopathic hypopara will benefit. Those with idiopathic hypopara who have the autoimmune form, that would be getting back to anybody with any kind of an autoimmune form where we'd have to also tolerize you to the parathyroid hormone so that you didn't have an immune rejection of the cells that were put in.
Okay, and then this is a big question. What is the timeline for your research being available to our patient population?
You know, if I knew the answer to that question, I would tell you. My guess is it's not going to take as long as it's taken to get pancreatic beta cells to the clinic, because they ended up solving a lot of the problems that we are now going to be able to piggyback on their success. So my hope is once we get these PTH protein forming cells to function in mice, and maybe that would happen in the next 3 years or so, that this would then have to go to a company for much, much more development, just like ViaCyte has taken over the pancreatic beta cell development. And then it would be another 3 to 5 years. So I'm saying 5 or 6 years, that would be optimistic, but that would, here's my concern. I'm a stem cell biologist, so I always hear people give promises and the promise of stem cell research. And what I don't want you to hear is that I am promising you that this is going to be ready in 5 to 6 years, because then when it isn't, then the promise wasn't kept. So I can tell you that I optimistically hope that within 5 to 6 years, this could get to phase 1 clinical trials, but I don't know.
Okay, and I have one more question. If you have a transplant of your gland into your arm, can your body reject it?
Not if it's from you, your patient. If it's autologous, if it started, that if it was derived from your own blood cells or your own skin biopsy at the beginning, where those cells were then induced to become pluripotent stem cells and then differentiated into parathyroid, that should not be rejected because the DNA in those cells is your DNA. If we did a third party, if there was like a bank of parathyroid cells that came from somebody else's cells, and those were what we used, then there would have to be some way to make it so you were tolerant and your immune system didn't reject it. Either you'd have to be on some kind of immunosuppressant drugs, or I know one of the things they're working on with the pancreatic beta cells, and again, we can piggyback on this, is a way of encapsulating them so that they function. They can sense, they'd be able to sense the calcium and secrete the parathyroid hormone, but that they'd be shielded from your body's immune system somehow so that they wouldn't be rejected.
Okay, now we have a comment here. I think I speak for the entire community just to say an enormous thank you for everything you are doing and for the hope that you have given us. Also, great lecture. So happy to have Dr. Krause as part of our conference now. Thank you for your incredible work.
Well, thank you. That's not a question.
I wanted, and that wasn't a question. That was just a statement.
Right.
One quick question. If you still have parathyroids in your neck, but your PTH level is undetectable, will you be able to take advantage of stem cells?
Yes, I don't know why if you have parathyroids that are still in your neck that they're not functioning. But let's assume that they're not functioning because they were affected maybe by prior surgery so that they didn't survive as functional cells. I don't know the answer exactly for this person's question, but I don't see why, if we develop them in vitro and from stem cells, that why they wouldn't work when they were implanted into the arm.
Okay, well, you provided us with so much information, and it's nice to know that there is a future, and we appreciate all of your work and everything. And I'm so glad that I found you on Twitter, and that's how we connected. And I know people have been asking how to connect with us here. On Twitter, it's at HypoPARAthyroidism, one word, and on Instagram, HypoPARAthyroidism. So thank you, Dr. Krause, and thank you everyone for being part of the first session today. And we will see you back here at 9:15. Thank you, Dr. Krause, very much. I appreciate it.
Thank you.